This what?
Yeah. So,
This what?
yeah. So, welcome, guys, back to Navigating Nano. This is season 4. Very excited. We have a distinguished professor, Dr. Afono from
Afonin.
Afonium. Close. Okay. Sorry. I was really trying my hardest before you got here. But yes, this is season 4. He's all the way from UNC Charlotte, and he has some exciting things to share with us. And so, to begin, my first question usually is like an icebreaker. For people that don't know, can you share a little bit about your background and what led you to start studying your career in your field of expertise?
Sure. So I'm originally from Russia, from Saint Peterburg. So I went to school there, to the university there. So I got my master's degree there in chemistry. And while the last year of the university, some guy from Ohio, from Bowling Green State University, some professor from there came to recruit graduate students. And I was asked to drive him around. Okay, so I was driving him around and he asked me if I want to try to fill out the application and do some simple tests. So I did the test. He got a check right away, said that I'm accepted already. So he invited me for a PhD. I was not planning at the beginning to go for a PhD program, but then I was thinking, why not? So I came, and I was particularly interested in biological research, biochemistry, because I was working in Institute of Molecular, I think it's called Molecular Biology and Genetics, if you translate it literally. So I was really interested in how life works and how we can influence different biological processes. So back then I worked with the protein infections, prions. It was a hot topic back then. And then when I came to States, I wanted to continue working in this way with the biology-related problems. So one of the professors at the department that I joined, and it was Center for Photochemical Sciences at Bowling Green State University in Ohio. So everyone did photochemistry there, organic chemistry. But there was one guy who did biochemistry, Dr. Neoklis Leontis, professor, who unfortunately died in the car crash recently. So it was a big loss for the society and for the community. He was a great leader and scientist. So, and he one of his characteristics was that he was very, very enthusiastic about the subject, and his enthusiasm was very contagious. So when he introduced me to the topic, and he worked with RNA molecules. I didn't know much about RNA, just basic school stuff, you know. And he basically laid out all these possibilities for me, explaining how this polymer is versatile and that we all really are what we are because of RNA, that all the complexity is coming from the complexity of our RNA that is produced. And the research seemed very interesting, and they just started this field of RNA nanotechnology, maybe 10 years before, not 10 years, but maybe 5 or 7 years before that, and he was one of the founders of the field. So for me, it was very intriguing that you can build nanostructures that never existed before in nature, but nature can see them as their own. It was pretty cool, so you can actually trick the nature this way. But we didn't do any cell culture work, it was all theoretical. So when I got my PhD, it took me 4 years to get it. I decided to move a little bit further and learn more about this process. So I went to Santa Barbara, UCSB, for my first postdoc, where I did it with Luke Jaeger, who was also a great scientist. He's a great scientist, he's still there. So I learned a lot more about RNA because Luke is he's an artist pretty much. Anything you want, he can do out of RNA. So one of his research, he did some heart out of RNA molecules, which is he's truly inspirational and talented scientist who understands this biomolecule very well.
Wow.
And then I wanted to take it further to the disease state. And I joined National Cancer Institute, wanted to stay with the government, but then at some point decided to try my luck with academia, got offer from UNC Charlotte, came, set up my lab, and here we go. That's why I am. So stayed in the same field pretty much.
It's interesting how you almost never end where you start. It's so many twists and turns. So that's interesting to hear. So you mentioned how you came from Russia, right? What advice would you give to other students interested in international studies like yourself?
Well, go for it. Yeah, especially if you have this opportunity, if you're not bound by any obligations, like family obligations and things like that. Back then I was not married, and I could just explore a little bit and invest in myself, get extra education. So if you have this opportunity, I think it's great, because the more you learn, the more cool and exciting things you can do with your life. Basically, the further you go, the more doors become available to you, windows of opportunities, so to speak, right? So you can, like when you graduate, you have just 2 or 3 doors that you can choose between, right? But if you go further, this number grows exponentially. And then you can truly choose the job that fits you the most. And basically, the best job is the job that you love, because then you don't work. There is a saying that if you find a job that you love, you will never work a single day. I think my job 100% fits this definition. So if you really like what you do, you never work. And you work all the time at the same time.
I agree. You never know, especially as a researcher, one conversation can lead to so many different opportunities, So never shy away from international studies. I went to Thailand before, and it changed my whole perspective.
You went where?
Thailand.
Thailand? Okay.
Yeah, that's the only place I've been out of the country, but it was
so far. Yeah.
Fascinating. Yeah, so far
the whole
it was
world is yours.
You get to see all the different learning styles and how they teach their students, and all they focused on was test and taking and things like that. So I'm not the best test taker, so that kind of opened my eyes a little bit, but it was interesting to see. Like I say, international studies definitely is
yeah, international experience is always cool.
Yeah, I agree.
And especially if you speak the language of the country where you go.
We were supposed to learn the language. I didn't pick it up, but it was a learning experience.
Maybe second chance. You can get a second chance one day and go there again.
Hopefully. So yes, I don't think you mentioned this, but you are the founding member and vice president of the International Society of RNA Nanotechnology and Nanomedicine. Can you share a little bit about that?
Yeah, so the field of RNA nanotechnology, as I mentioned, is relatively new, to the world. It's about 20 years old or so. And but it's very much appreciated in the community. So more and more scientists were starting doing similar research to mine. So more and more people realized the potential of RNA and wanted to apply those technologies to the problems that they were pursuing before. So the field started growing, but it was not kind of unified in the way. And that was the main motivation for the society, and the main driver and organizer, so to speak, of the whole process was Dr. Ph.D, who is a professor now at the University at Ohio State University. So Dr. Guo, he's also a pioneer of the field. He basically started the therapeutic RNA nanotechnology part. So he came up with clever ideas of how those particles can be used to deliver different therapeutics in human cells and in animals and so forth. So he was very inspirational for many other groups. So he was kind enough to invite me to participate in this process, and it was quite a challenge at the beginning, but it was fun, a lot of fun. We were having a lot of fruitful conversations, discussions where the field should go, what the mission should be, where we see ourselves in 10 years and so forth. So just to give you some update, currently we are establishing the Society Journal, which is going to be called RNA Nanomedicine. Thank you very much, RNA Nanomedicine. And we already have the journal in place and have, I think, 5 to 10 submissions.
Wow.
So soon we will make it public announcement, and it will be available for everyone to submit their research. But now we have a society, we have a journal, so obviously it speaks a lot about the field and how it's growing, and hopefully it will just continue going this way, full speed.
Okay, so we do have listeners from science background and from non-science background. So can you explain what RNA is exactly?
So RNA, yes.
Some of the technologies that you can make from RNA.
So RNA, if you look at this, it's just a biopolymer, which is composed of 4 main building blocks, 4 units, okay, which are combined to form a single chain, okay, to form a chain. But depending on the order how those units are combined, the function of this chain will differ. But RNA mostly known as the intermediate between the DNA and protein. So if you go back to school and remember this central dogma of molecular biology, you see that RNA goes to DNA, DNA goes to RNA, goes to protein, so it's a flow of the genetic information that all living cells have, okay? And then a protein was also thought to be the key player in the life. DNA would always be considered as the storage of the genetic information, and RNA was just a simple intermediate. But in fact, it's not quite true, because RNA is what defines our complexity and orchestrates all the processes, biological processes within the cells and body. Because if you look at the number of proteins or open reading frames that code for proteins and compare them between humans and, for example, flowers, you will see that we have about the same number of proteins, but the complexity is much higher in our case. So this complexity is coming from the amount of RNA that is being transcribed, produced from DNA, but not translated into the proteins. Okay, so we have some RNA that is produced, but it doesn't code for any protein at all. Okay, so the amount of this RNA defines and regulates lots of processes, biological processes, participate in development of diseases also. Therefore, understanding the way RNA works would help us to deal with certain diseases, with certain pathogens, and it also makes RNA a very attractive material for therapeutic applications, since it's so versatile functionally. Understanding the function of RNA can help us to regulate it in the way that we can cure something, for example. But it also opens a possibility for RNA nanotechnology, because now we can use this material to build some structures that can be recognized by the cells as their own. Okay, so we can hack, so to speak, we can hack some biological processes already by using this polymer. Okay, but yeah, it's essentially a biological polymer, biopolymer, that is highly functional on its own. Okay, if you take DNA, it's very inert functionally, but RNA can by itself, without help from any protein, perform some useful tasks. It can catalyze reactions, for example, induce certain immunological responses by interacting with proteins. It can interact with other RNA molecules and promote their activation or deactivation and so forth. So it's a very, very biologically and structurally functional versatile biopolymer.
Yes. So as you guys hear, RNA is very important to understand the body holistic. They usually refer to RNA as like the junk DNA.
Junk DNA. So
yeah,
junk DNA would be referred to the DNA that is not coding for proteins.
But now we know it regulates diseases.
But now, yes,
systems.
it's not junk at all.
Right.
So it's basically, yeah.
Thanks to you guys like you. So
without this junk, we wouldn't be who we are.
Yeah, this is true. Okay, so for my next question, the first time I heard about RNA was like around COVID time, right? It's talking about like mRNA vaccines and things like that. What applications do you have for listeners that might not be aware in terms of like drug delivery? Because I know you just gave a talk about that. Can you share a little bit about that with the audience?
Right, so if you think about mRNA vaccines, in the way it's also can be considered as RNA nanotechnology, okay? Because there is a lot of work and rational design implemented in those vaccines, because you have to make those RNAs to be stable enough, to not degrade very fast, to produce enough protein, to not be recognized as foreign RNA and induce some undesired immunological response, okay? It has to be delivered into the cell, because RNA is negatively charged biopolymer. It's a large negatively charged molecule which wouldn't go through the membrane, okay? Cell membrane, lipid membrane. So you really need to implement some delivery aspects to this work. You have to program your proteins within this sequence, and again, keep this sequence non-immune stimulatory by itself, but also highly translatable. And this is just one of many applications. It's a great technology. It's highly tunable because you can tune it against any protein or changes within the protein that virus can implement later on, so you can adjust your vaccine to new threats if you talk about mRNA vaccines. But there are lots of other applications that RNA can play. There are several FDA-approved drugs which are based on RNA interference, which is another Nobel Prize-winning technology that was discovered at the end of last century, I think 1998, if I remember correctly, by Fire and Mello. They discovered that RNA, double-stranded RNA, if introduced into the cell, and if this double-stranded RNA has a certain sequence, which is complementary to the sequence of some parts of messenger RNA within the cell, then this double-stranded RNA can employ help from some proteins and guide them to find this messenger RNA sequence to destroy it or attenuate its function. Okay, it's a very powerful technology because now you can basically turn off any translation within the cell on condition. Another important Nobel Prize-winning technology is the CRISPR-Cas9 technology, which allows you to, for example, do gene editing. So if you have some diseased genes that you want to fix, potentially, this technology can allow you to do this. And what is interesting is that this technology is also guided by RNA. Right. The work is done by protein, but the guiding instructions to which DNA and where should be modified or cut is coming from the sequence of RNA that again you can program and synthesize. Okay, so you can hack this process and make it very useful for various applications. And there are a lot of other processes that you can also take advantage of, biological processes. And also there are some artificial ways to introduce new classes of RNAs, as I mentioned before. There is a powerful process called SELEX, systemic evolution of ligands by exponential enrichment, which is ability of scientists to select experimentally a completely new strands of RNA that would recognize specific targets of interest that you specified before. So you have a molecule that you want to capture or bind to, so you can use SELEX to produce an RNA molecule or DNA molecule also. It can work for both biopolymers. That will specifically bind to this protein only with the high affinity, high selectivity, and this can be used in place of antibodies, for example, okay? Monoclonal antibodies. So because the sequence will always be the same, there will be no batch-to-batch variability. The binding affinity is not quite comparable, but can be probably improved, especially if several aptamers can be put together. And I showed you an example with the anticoagulants later on, which takes advantage of aptamers. But again, those aptamers were DNA-based aptamers. But the idea is absolutely the same. So this is a completely different opportunity that would not be possible if not the group of several talented scientists who discovered this process.
Yes, we thank you, scientists.
Yes, thank you.
It's almost like you're making like RNA robots in a way.
Yeah, you can make an RNA robot. So you can make someone to make your work. So that's another actually good aspect. So what my lab is working on is to make conditionally activatable devices. So you can make smart nanoparticles that can enter the cell and based on the cellular environment make a decision about execution or not execution of certain functions. For example, if it enters the disease cell and finds some disease marker there, it will kill the cell. But if the disease marker is not present there, it will be just degraded and the cell will survive. Okay? Yes. So that's a nanorobot, so it can think. It's a very simple, it's it's a very simple operation. So it's basically AND logic gate. So if you have this present and this present, then you will have a positive outcome out of it. But it's it can be expanded much further, and you can actually program much more complex ways of computing within the cells.
Thank you, sir. Thank you. So now we're going to turn it over to the audience for the sake of time to ask you guys questions. So whoever want to go first, please feel free. We're listening. Yes.
What's the biggest RNA that you use in your study?
Yeah, so the longest, you mean, the longest RNA. So we work with the short sequences. So we try not to exceed maybe 100 nucleotides long for the single strand. But normally we work with even shorter strands than this, because sometimes we prefer to chemically synthesize them, and there are some limits to what you can synthesize chemically. We use transcription mostly, but yeah, we don't work with too long strands because there's no application for them in our research.
How do you track the DNA or RNA nanoparticles inside the cell?
Mm-hmm. So that's a challenging process. So we normally use fluorescent tags, but then how do you know that you have a fluorescent tag which is still attached to your particle, and you are not just tracking the fluorescent tag on itself, right? So we use combinations of fluorescent tags. We have several tags of different colors present on the same particle, and if both of them are colocalized, or if they can produce some Foster's resonance energy transfer between them, that means that they're still in close proximity. It means that most likely, at least those strands to which they're attached are somehow still connected to each other. But because our assemblies are multi-stranded, the ideal would be to have as many dyes as number of strands that you have. So if you have 5 strands, it would be nice to have 5 dyes and localize all of them. But yeah, we use 1 or 2 dyes. 2 is the most for now.
Thank you.
Nice.
Hi, I'm Parker. Are there any roadblocks that you feel like other researchers may be ignoring or not cognizant of in your field? And what would be your message to help prevent people from getting lost in like rabbit holes you perhaps maybe already made it out of?
Yeah, so I think that there are some, there are many different roadblocks, and we can start with just education. I think RNA as a pedagogical tool is underused, underrepresented in school education, at university level, and so forth. So lots of graduates, even from biology departments, for example, they have very vague ideas about the diversity of RNA functions, structure of RNA, its ability to fold on itself and dictate certain function through this. So I think this would help a lot if we introduce more specialized classes which would just focus on RNA and RNA technologies. And by doing this, we would produce a much more competitive and diverse and needed workforce, because obviously the field is blooming, but I think one of the reasons why it's not at the full speed is because we just don't have enough RNA scientists, RNA biologists. RNA is the hard molecule to work with to begin with. So people prefer to avoid working with this if possible, if they can substitute the need with some other options. But I think it's just because the training is not there. Once you learn how to work with this and pass this knowledge downstream, I think we will address a massive lack of the RNA scientists and address the demand on this. This is one of the fields. Another thing is that if you want to do some clinically relevant work, you really need to understand the problems, the current problems. So we need more working groups where we would connect RNA scientists with some regulatory agencies such as FDA, for example, or some pharmaceutical representatives, so we can work together to identify some solutions for the common goals. And there are many other things, but I think with time it will be addressed. And I think that the fact that we had this mRNA vaccine frontrunners a few years ago really helped to bring attention of the society to RNA. But I don't know also how many people got scared even more because of this, because there is a lot of controversy, right, and people opinions about these approaches, which I think it's also is because of the lack of the education in this area, which again has to be promoted at the college level, but also at school level. So my lab would do some outreach with the high schoolers, with the undergraduate, graduate students, and our university, now in other schools also we host different faculty, even school teachers, and we try to explain them the notion of RNA and related technologies better so that they can convey it. You basically teach one teacher, you teach 100 people, right? So it's that might be a good approach.
Yes.
In what ways do you foresee that RNA nanoparticles contributing to the development of smart bio materials for therapeutic application?
Yeah, so it's basically learning from nature, because nature does it already. RNA is a dynamic polymer that can activate or deactivate certain processes at right times based on different mechanisms. So many different mechanisms. So what we need to do is to understand those mechanisms and then think about the way to implement them in our nano design and then test them, okay? And that's what we do basically. So I think that creation of smart personalized therapeutics using RNA molecules is quite possible. It will require some effort, but it's very possible to create some very robust design principles that will work steadily and will be implemented against multiple different diseases and abnormalities. Okay.
Nice. Any more questions from the audience?
I'll knock it out one more. In terms of your future research goals, like what would be your dream to accomplish or prove before you retire? And are there any new fields you see emerging from your field you're excited to see explored in the next generation?
Well, the dream, of course, is to leave a good footprint, okay? As an educator, so make sure that we produce high quality researchers that can make a difference in the world, okay? And so far I've been lucky that all my students have been awesome and great, and they all chose the career paths that fit their expectations, and they were able to accomplish a lot throughout those processes, and they now disseminate the knowledge and expertise and experience further on. So that's the big goal. So obviously leave a good mark and make a world a little bit better place in terms of RNA technologies, but also, of course, to create some either develop a technology that would contribute to the creation of certain or development of certain drugs, therapeutics, or develop a therapeutic and bring it to clinical trials. Either way is fine. Another way is important, because you can just come up with the concept that later can be applied by someone else, but you will know that at least you put your input was also taken, okay, in this big project. So I think that would be good enough for me. So if any of the technologies or ideas that we share through publication or conferences were picked up by someone and resulted in some therapy, I would be very happy with that. All right? Yeah.
Any more questions? All right. One more. Okay. This could be our last question.
How do you address potential challenges related to stability of RNA, the RNA nanoparticles delivery in vitro and in vivo, especially given the susceptibility of RNA to degradation?
Yes, excellent question, and it's actually it's not necessarily a bad thing that RNA is non-stable. There is a reason why RNA is non-stable, right? Because it has to be present or non-present within the cell up on demand. Okay, so when it's not needed anymore, it has to be removed out of the game. So it's a It's a very clever mechanism if you think about it. Therefore, I don't think we should get rid of this. You can easily get rid of this by introducing chemically modified nucleotides, as I showed in my presentation. You can make RNA nanoparticles be super stable in blood without any carrier attached. So it's a naked nanoparticle and it can last for hours in blood serum. But I think the beauty of this technology is that you have to make it to live long enough to perform the function you wanted to do, but then kind of get dissolved or get digested completely after that, so it doesn't do any additional harm. So there will be no accumulation, no side effects. So this timing is very important, and I think the timing can be regulated by implementing certain structural parameters into the nanoparticle rather than chemical modifications. So it can still be biocompatible RNA with a prolonged lifetime. Okay.
Thank you.
All right, no more questions? Okay, I do have another question.
Okay.
I cut it short because I wanted to make sure the audience got their questions asked, but this is kind of similar to one of the audience questions. So with RNA, I know a lot of its application is like with vaccines and things like that, but do you see it being used in things other than biological, like maybe a new material feedstock?
Yeah, so for new materials, the instability, chemical instability can be a challenge.
Yeah.
Okay, but you can make new materials which will be operational inside the cells. So they don't necessarily should be therapy related, but you can build some responsive devices, for example, that will react to some environmental stimuli and inform the researcher or the cell or whatever you want about those changes. So you can use them in sensing, in biosensing, okay? Which is also not necessarily therapeutically related, but rather maybe research oriented. So there are some research tools where RNA definitely can be used and is used. And again, it's not therapy, but it was basically to explain better the biological processes and understand better different pathways and so forth. So RNA is a very potent tool also. Okay, it's just a tool which can be applied for various applications, for many different applications, but probably mostly biologicals because again of the stability, of the cost, overall cost. The shelf life is not that great also, but you can optimize, of course, the shelf life also of RNA. But yeah, that would be my take.
All right, well, we are approaching the end of our podcast. Yes, thank you for sharing all your expertise on RNA. We have a tradition here where the previous guests leave a question for the current guest, and then you do the same. So the previous guest said, some people question the need for a college degree. What is your response?
I think it should be a personal choice. It should not be enforced, definitely, because, well, it's expensive in many cases, and if you don't really need it, it definitely helps you to increase the level of your awareness and understand the world around you better. Okay, it's a universal degree, so it just helps you to understand the world around you better and maybe fit better into the society. But you can do the same now by accessing all these great learning tools online. It's all about motivation, because if you come to the college and you are unmotivated, maybe you can argue that the college can help you to somehow force you to get some knowledge in, right? But I think a motivated person can definitely educate him or herself at home. But college definitely helps because it just guides you through this process.
Yes.
But I don't think it's a requirement. It shouldn't be a requirement. It's only if you need it. But it also seems like with the modern technologies and with the modern lifestyle and the dynamicity and the amount of information that you get on daily basis, any additional education, whether it's college or homeschooling, will definitely help you to fit better in. Yeah.
Well, thank you, sir, again, and thank you for your contribution
Thank
to
you.
RNA discoveries. we're excited to see where you take this in the future.
Thank you.
Yes.
All right.